Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi dan salam sejahtera untuk kita semua. Perkenalkan, saya Nindya dari Swantiningsih. Saya adalah dosen di Fakultas Kedokteran Universitas Airlangga yang ditempatkan pada Departemen Mikrobiologi Kedokteran. Untuk latar belakang pendidikan, saya menyelesaikan pendidikan profesi dokter di Universitas Airlangga pada tahun 2010. Kemudian saya menyelesaikan pendidikan Master of Medical Science in Immunology di Harvard University di Amerika Serikat pada tahun 2016. Saya mengampu beberapa mata kuliah. Salah satu mata kuliah utama yang saya ampu bersama tim teaching adalah mata kuliah mikrobiologi kedokteran. Mikrobiologi kedokteran ini adalah mata kuliah yang wajib untuk mahasiswa pendidikan profesi dokter yang ditempuh pada semester ketiga. Pada mata kuliah mikrobiologi kedokteran, mahasiswa akan memperoleh pengetahuan yang komprehensif terkait dasar-dasar mikrobiologi kedokteran, pengetahuan mikroba yang menjadi penyebab penyakit infeksi, baik bakteri, virus, maupun jamur, serta pengetahuan mengenai imunologi penyakit infeksi. Selain itu, mahasiswa juga akan diajarkan keterampilan melakukan pemeriksaan untuk menegakkan diagnosis penyakit infeksi. Setelah menempuh mata kuliah mikrobiologi kedokteran, capaian pembelajaran yang diharapkan adalah mahasiswa mampu menjelaskan pengetahuan mikrobiologi kedokteran secara komprehensif. Mahasiswa akan mampu menjelaskan dasar-dasar mikrobiologi, mikroba yang menjadi penyebab penyakit infeksi, baik itu bakteri, virus, maupun jamur, serta mampu menjelaskan aspek imunologi dari penyakit infeksi. Selain itu, mahasiswa juga diharapkan akan memiliki keterampilan untuk melakukan pemeriksaan guna menegakkan diagnosis dari penyakit infeksi. Salah satu topik yang saya ampu dalam mata kuliah mikrobiologi kedokteran adalah interaksi antara host dan mikroba. Perkuliahan ini diberikan pada mahasiswa pendidikan profesi dokter di kelas internasional. Pada perkuliahan ini, mahasiswa akan dijelaskan mengenai 3 pokok bahasan, yakni pertama, jenis-jenis interaksi host mikroba, kedua, tahapan interaksi host dan mikroba, yang ketiga adalah outcome yang mungkin terjadi akibat adanya interaksi antara host dan mikroba. Hal ini akan membantu mahasiswa untuk mampu memenuhi capaian pembelajaran dari mata kuliah mikrobiologi kedokteran pada aspek mampu menjelaskan pengetahuan dasar imunologi penyakit infeksi. Perkuliahan ini kami lakukan secara daring dengan metode ceramah dan diskusi. Contoh kasus diberikan pada awal perkuliahan untuk menarik minat mahasiswa. Pada akhir perkuliahan, contoh kasus akan didiskusikan. Hal ini akan memungkinkan terjadinya interaksi yang lebih intens antara dosen dan mahasiswa maupun antar sesama mahasiswa. Pada perkuliahan ini, ada inovasi yang kami lakukan untuk menunjang perkuliahan. Inovasi yang kami lakukan adalah kami selalu berusaha menyertakan contoh kasus di dalam setiap materi perkuliahan. Contoh kasus kami berikan untuk membantu mahasiswa lebih mampu untuk mengaplikasikan materi perkuliahan dengan kasus sehari-hari yang ada di lapangan. Contoh kasus didiskusikan pada akhir perkuliahan. Hal ini akan meningkatkan interaksi antara mahasiswa dengan dosen dan antar sesama mahasiswa juga diharapkan akan mampu meningkatkan kemampuan presentasi dari mahasiswa. Terkait sistem penilaian mata kuliah, pada mata kuliah mikrobiologi kedokteran, penilaian didasarkan pada gabungan antara ujian tulis, tugas perkuliahan, dan soft skill. Yang dengan bobot masing-masing yaitu 30% UTS, 30% UAS, 30% ujian teori praktikum, 5% tugas perkuliahan, dan 5% soft skill yang dinilai dari kedisiplinan, keaktifan, kesopanan, dan kerapihan. Setelah ini, saya akan menyampaikan perkuliahan dengan topik interaksi host mikroba yang diberikan pada mahasiswa pendidikan dokter kelas internasional.
Good morning, everyone. Assalamualaikum warahmatullahi wabarakatuh. I'm Ika Kadariswanti-Ningsih. Today we will discuss about the host microbe interaction. After completing this lecture, we hope that you will understand the fundamentals of human microbe interactions and their implication for both of living entities by understanding various types of human microbe interaction and then general stage of human microbe interactions and also the possible outcomes of infections. And for today's lecture, the outline will be types of the human microbe interaction, and then we will discuss the general stage of human microbe interaction, including encounter, colonization, entry, invasion, and dissemination, and also possible outcomes of infections. So we'll start with this case study. A 71-year-old man was treated for right lower extremity cellulitis with a 10-day course of antibiotic cephalexin. A few days after completing the course of antibiotics, he started having loose and watery diarrhea. A bacterial culture of the stool was negative for Salmonella, Shigella, Campylobacter, Yersinia, and also Vibrio species, but a PCR assay for Clostridium difficile toxin B gene was positive. And the question is, how did the patient develop diarrhea caused by Clostridium difficile? I hope that after completing this lecture, all of you can get the answer. The interaction between humans and microbes are very complex and far from being completely understood. What is known about the interaction between these two entities play an important role in the practice of diagnostic microbiology and in the management of infectious disease. Understanding this interaction is necessary for establishing methods to isolate specific microorganisms from patient specimens and for developing effective treatment strategies. So we will start our today's lecture with types of human microbe interactions. Symbiosis is the relationship between two dissimilar organisms living in close association with each other. So we can call the interaction between two dissimilar thing organisms who live in close association with each other as symbiosis. And generally, there are three types of symbiosis: mutualism, commensalism, and parasitism. The type of symbiosis is defined based on the benefit or harm received by each organism. For example, in the mutual symbiosis, both of the organisms receive benefits, but in parasitism symbiosis, one receiving benefits, but the others is receive harms. For example, is all of the pathogen bacteria that cause disease for human. And next, we will talk about the general stages of human microbe interactions. The complex relationship between human hosts and medically relevant microorganisms are best understood by considering the sequential steps in the development of host microbe interaction. And the stage of interaction include first is physical encounter between the host and microorganism, and then two, the colonization of the microorganism on the host surface, and three, the microorganism entry, invasion, and dissemination, and the last one is about the resolution of the outcome. So we will start with the first stages, which is encounter. So because microorganisms are found everywhere, human encounter are inevitable. Which microbial population a human is exposed to and the mechanism of exposure are often direct consequences of a person's activity or behavior. Certain activities carry different risks for an encounter, and there's a wide spectrum of activities and situations over which a person may or may have not absolute control. For example, acquiring salmonellosis because fails to cook the meat thoroughly is avoidable, but contracting tuberculosis as a consequence of living in condition of extreme poverty and overcrowding may be unavoidable. So humans encounter microorganisms when they enter or are exposed to the same environment in which the microbial agent live, or when the infectious agent are brought to human host by indirect means. The environment or place of origin of the infectious agent is called reservoir. And microbial reservoir includes humans, animals, food, water, air, or soil. The human host may acquire the microbial agents by various means, referred to as the modes of transmissions. So the mode of transmission is direct when the host contacts the microbial reservoir, and is indirect when the host encounter the microorganism by an intervening agent. The agents of transmission could be living entities, which we call vectors, such as animals or other humans, and it also can be vehicles, which is a non-living thing. And humans play a substantial role as microbial reservoir. For example, the passage of a neonate from the sterile environment to the mother's womb through the birth canal, which is heavily colonized with various microbes agent, is a primary example of one human directly acquiring microorganism from another human. Other examples in which humans serve as microbial reservoir include the acquisition of streptococcal pharyngitis through touching, hepatitis through blood transfusion, gonorrhea, syphilis, and AIDS through sexual contact, tuberculosis through coughing, and common cold through sneezing. Here is the list of several modes of transmission. So you can see other indirect modes of transmission through vehicle or through vectors. So the next step after encounter is colonization. Once a microbe attains contact with a human host, the outcome of encounter depends on what happened during each step of interaction, beginning with colonization. The human host in microbial colonization defined as persistent survival of microorganisms on a surface of a human body. And it is dictated by the defense that protect vital internal tissues and organs against microbial invasion. The first defense are the external and internal body surface that are in direct contact with the external environment and are the anatomic regions where the microorganism will initially come in contact with the human host. This surface includes skin and also mucous membrane that lining the mouth or oral cavity, the respiratory tract, the gastrointestinal tract, and also the genitourinary tract. So the first host, the colonization from the host perspective, the first one, we will talk about the resistant factors, in this case, skin. So skin serves as a physical and chemical barriers to microorganisms. Its protective characteristic can be seen in this. So it has outer layer, it has several glands, and also it has epithelium and skin-associated lymphoid tissue that each of them have several protective activities against microbe. The next resistance factor is mucous membranes, because cells that line the respiratory tract, the gastrointestinal tract, and also genitourinary tract are involved in numerous functions besides protections. They are not covered with a hardened and acellular layer as is skin surface. So it's covered with mucous membranes that also has several components to protect them against microbes, such as mucosal cells, goblet cells, and mucus-associated lymphoid tissue, that you can get the protective activity from each of them from this table. And besides the general protective properties of mucosal cells, the mucosal lining throughout the body have characteristics specific to each anatomic sites. For example, the stomach has a lower pH, and then the small intestine has bile salts and also peristaltics, and then vagina has low pH, and bladder has flushing action of urine. So it add more protective activity against microbe. So now we will see the colonization from the microbes perspective. So as previously discussed, microorganisms that inhabit many surfaces of the body, we refer them as colonizer or normal flora or normal microbiota. And collectively, we can call them human microbiome. Some are transient colonizer because they are able to survive but do not multiply on the surface and are frequently shared with the host cell. And others called resident microbiota that not only survive but also thrive and multiply, and their presence is more persistent. And each body site has different composition of normal flora. Here is some of the microbes genera that we consider as the normal flora of each anatomic site. So colonization may be the last step in the establishment of a long-lasting and mutual beneficial or harmless relationship between a colonizer and the human host. Alternatively, colonization may be the first step in the process for the development of infection and disease. Whether colonization will result in harmless or damaging infection, it depends on the characteristic of host and the microorganism. In either case, successful initial colonization depends on microorganism ability to survive in the condition first encountered on host surface. So here are some of the microbial factors that contribute to colonization of host surface. For example, usually microbe will search for moist area. They will be located in the moist area that make them have more chance for survive. And maybe some of the microbes such as Staphylococcus has a salt tolerance activity, so they can survive against like our skin conditions. Some of them also have these factors that can make them have higher chance for survive. And then the next stage of human microbe interaction is entry, invasion, and dissemination. So in most instances, to establish infection, microorganisms must penetrate or circumvent the host physical barriers, in this case, skin or mucosal surface. And overcoming these defensive barriers depend on both host and microbial factors. When these barriers are broken, numerous other host defensive strategies are activated. So here are some factors that contribute to the disruption of skin and mucosal surface of the host, in this case human, such as trauma. Trauma can be intended trauma, such as surgical wounds. So like we make an incision in the patient, and then it can be inhalation, and then diseases or overuse of antibiotic. All of them can cause disruption of the skin and mucosal surface of the host that can make microbes easier to entry to the host. Once surface barriers have been bypassed, the host responds to microbial presence in the underlying tissue in various ways. Some of these responses are non-specific, because they occurs regardless of the type of invading organism. So one of the non-specific response is through phagocytes. So when there are some bacteria, so phagocyte will go and endocytosis to the bacteria, and it will form the phagosome, and phagosome then will be fused with lysosome and then create the phagolysosome. And after this, there are still three possible outcomes. One, the bacteria can be survive in the phagocytes. So it can become a long-term survival of bacteria in phagocytes. And two, bacterial destruction. So phagocytes can destruct the bacteria into bacterial fragments. And the last one, the last possible outcomes can be destruction of the phagocytes. So bacteria can multiply. So the previous one is like an innate immune system against bacteria. Next, we also have a specific immune system. In this case, adaptive. We also call it adaptive immune system through our B and T cells. So when B cells with its antigen receptor can get the microbial antigen, so this will activate the B cells, and it will go to clonal expansion, and then these B cells will also present it to the helper cells that will help B cell to make more B cells. It will stimulate B cells to undergo maturation and plasma cells for making more highly specific antibody and switching from IgM antibody to IgG antibody production, and also produce the memory B cells for the next encounter with this kind of microbes. And the next adaptive immune system is our T cells. So when T cell with its antigen receptor meets with some antigen presenting cells, such as macrophages, B lymphocytes, or dendritic cells that already have antigens on its cells. So it will activate the T cells, and activated T helper cells or also T cells will help to eliminate the microbes through several actions. For activated helper T cell, it will increase its number and then release cytokines and stimulate, and these cytokines will stimulate another phagocytes and NK cells and other component of inflammation, and assist B cells in antibody productions, such as our previous slides. And then for activated cytotoxic T cells, when it gets activated, so it will increase its number and also target and destroy host cells that are infected with microorganisms. So in the entry, invasion, and dissemination from the microbes perspective, virulence factor provide microorganisms with the capacity to avoid host events and damage host events, and it will damage host cells, tissues, and organs in a number of ways. Some virulence factors are specific for certain pathogen genera or species, and substantially different exist in the way of bacteria, viruses, parasites, and fungi cause this disease. And you will learn about this characteristic of the virulence factor of each microbe in the next lecture. Here are some of the virulence factors, such as pili or adhesion, and then antiphagocytic factor, such as capsule or slime layers, biofilm formation, and then toxins. And then next is about the outcome of infections. Basically, outcomes depend on the state of the host health, the virulence of the pathogen, and whether the host can clear the pathogen before infection and disease cause irreparable harm or death. So here are some of the host factors, and here are some of the microbial factors. So each of them will determine in which the outcome will be. It can be restoration of the host to the complete health. It can be restoration of the host to health with residual effects, or survival with host health, but host health severely compromised, or the outcome can be death. And the last one is the treatment of infection is often difficult and not always successful, because much of the damage may already have been done before appropriate medical intervention is provided, and microorganisms can too much a head start. So one of the best way is preventing the infectious disease, and it can be through several measures such as listed here. One of the most popular way is immunization and sanitation. We're already in the end of this lecture, so we'll close it with this case study. And the question is, how did the patient develop diarrhea caused by Clostridium difficile?
Hello. The question: when Clostridium difficile become established in the colon of individuals with normal microbiota, they can in the intestine primary bile acid exposure cause germination of Clostridium difficile, and then in the colon they can colonization and toxin production and damage to the intestinal mucus, and excessive mucus production result in diarrhea, colitis, and the formation of pseudomembrane. They have toxin production.
Thank you, Ayu, for answering. So you already answered about the pathogenesis of how Clostridium difficile caused the diarrhea. Thank you so much for answering. But my first question is, what kind of things here that cause the patients get Clostridium difficile in a high number in his intestine?
Maybe I try to answer. Because the diarrhea is because of antibiotic associated diarrhea, because of the use of antibiotic before. So the use of antibiotic can disrupt the microbiota in the intestine. So it becomes Clostridium becomes many. So then it causes toxin maybe.
Yeah, you are right, Navsu. Thank you so much for answering. So overuse of antibiotic can cause disruption of the skin, and in this case, especially, it can cause disruption in the composition of the normal flora in the intestine, and it can make the Clostridium difficile to replicate in very high number. Thank you so much for answering. Do you have any questions? Okay, thank you so much for today. And if you still have any question, please email me.
Demikian rangkaian video micro teaching yang dapat kami sampaikan. Terima kasih banyak atas perhatian Bapak Ibu sekalian. Wassalamualaikum warahmatullahi wabarakatuh.